Hi, welcome to Dawatini. The word mutagen is derived from the word mutate or mutation. So these are agents that permanently alter an organism's genetic material or DNA. That is, it can either be DNA or RNA. So mutagens are classified into three primary categories based on their structural properties and how they interact with the nucleic acid. So these three are chemical mutagens, physical mutagens, and biological mutagens. Let's have a look at chemical mutagens. These are foreign compounds that enter the cell and physically alter or mimic the structure of nitrogenous bases. The nitrogenous bases in question are adenine, thymine, cytosine, and guanine. The change usually leads to mispairing during DNA replication. The types of chemical mutagens include base analogues, alkylating agents, and deaminating agents. Let's look at base analogues. Base analogues are molecules that chemically resemble standard DNA bases so closely that DNA polymerase accidentally incorporates them into the growing strand. So we have an imposter who looks exactly like, say, adenine. So DNA polymerase, who is in charge of queuing the bases in a specific manner, incorporates the imposter where A is supposed to be placed. So a good example of this is the 5-bromouracil, shortly abbreviated as 5-BU. It resembles thymine but frequently shifts its structure to a pair with guanine instead of adenine. It causes a G to C transition or pair where the base pair would rather be A to T. So this is the mechanism of base analog mutation by 5-bromouracil, which is a chemical mutagen. On the strand, we have two nitrogenous bases following each other, that is A and T. So once in contact with the chemical mutagen 5-bromouracil, the result is thymine is substituted for 5-BU, that is 5-bromouracil. And as you can see, adenine remains adenine. In the first round of replication, we have the AT strand that maintains its integrity and the replicated strand that involves a bromouracil molecule. In the second round of replication, we still have the AT strand that maintains its integrity, and then we have the introduction of a guanine nitrogenous base. And the 5-bromouracil undergoes a tautomeric shift from keto form to enol form, or rather enol form. In the third replication round, guanine is finally paired with cytosine, and adenine maintains its 5-bromouracil base pair partner in its keto form. Replication results in an AT to GC transition. This is considered a mutation since it's an alteration from its original AT form. Let's now look at alkylating agents. Alkylating agents are chemicals that attach an alkyl group or a hydrocarbon to DNA bases, and this changes their hydrogen bonding patterns. An example is ethyl methane sulfonate, that is EMS, and methyl methane sulfonate, that is MMS. They add ethyl or methyl groups to guanine, causing it to mispair with thymine, as you can see on your right. Deaminating agents, on the other hand, are chemicals that strip amino groups from nitrogenous bases. A good and famous example of this is nitrous acid. It converts cytosine to uracil. It also converts adenine to hypoxanthine, which pairs with cytosine. And on your right, we have an illustration of the same. The first one results in a GC to AT transition mutation, and the other one results in a TA to CG transition mutation. So the third type of chemical mutagen is an intercalating agent. These are flat, planar molecules that physically slip between adjacent base pairs of the DNA double helix, and this distorts the structure. Once the structure is distorted, the polymerase becomes confused by the distorted shape and randomly inserts or deletes a base, and that causes a severe frameshift mutation. Examples of this include ethidium bromide, briefly abbreviated as ETBR, and acridine orange. Now we're done with chemical mutagens, let's go ahead and look at biological mutagens. So biological mutagens are living or virus-like entities that insert genetic material directly into a host genome, disrupting normal gene sequences. Common examples of these are transposons and viruses. Transposons are what people refer to as jumping genes. So an example of a virus acting as a mutagen is oncogenic viruses, that is, cancer-causing viruses. These integrate their own DNA directly into the host cell's genome. And an example is HPV, human papillomavirus, or hepatitis B. If the viral DNA accidentally inserts itself right in the middle of a tumor suppressor gene, the host gene is destroyed, leading to uncontrolled cell division, and this is what we refer to as maybe cervical cancer in the case of HPV. Now let's look at the famous jumping genes, that is, transposons. So mobile DNA sequences that can naturally cut or copy themselves out of one location in the genome and paste themselves into a completely different location are what we are referring to as transposons. When a transposon lands inside an active functional gene, it physically disrupts the reading frame, rendering that protein completely non-functional. In the image to your right, let's say the GTCA base pairs belong to a certain gene or are indeed a complete gene. They'll move from this section of the DNA strand to a different section. It's possible they will interrupt a functional gene where they will land. Therefore, when transcription is taking place, the result will no longer be in alignment with its original function. Now we're done with chemical mutagens and biological mutagens. Let's go ahead and look at physical mutagens. Physical mutagens are energetic forces that penetrate the cells to directly smash or rather break the DNA backbone or twist molecular bonds. The examples or categories that we're going to be looking at include ionizing radiation, non-ionizing radiation, and temperature. Ionizing radiation refers to highly energetic waves or particles that penetrate deep into tissue, strip electrons from atoms, and break chemical bonds. Examples include X-rays and gamma rays. They generate highly reactive free radicals inside cells and cause lethal double-strand breaks in the DNA backbone. Non-ionizing radiation, on the other hand, has lower penetration power and doesn't strip electrons but excites atoms. This causes them to form abnormal chemical bonds. An example of this is ultraviolet light, that is UV. It causes adjacent thymine or cytosine bases on the same strand to bond together, and this creates pyrimidine dimers. These dimers distort the DNA helix and halt transcription and replication. Guys, I'm so sorry about my cats. Non-ionizing radiation, on the other hand, has lower penetration power and does not strip electrons but excites atoms. This causes them to form abnormal chemical bonds. An example is UV, that is, ultraviolet light. It causes adjacent thymine and cytosine bases on the same to bond together, creating pyrimidine dimers. These dimers distort the DNA helix and halt transcription and replication. So the last type of physical mutagen we'll look at is heat, or rather temperature. Heat is a form of thermal energy. It acts by strictly increasing molecular vibrations. When the vibrations become too intense, they break the weak chemical connections such as the hydrogen bonds holding the two DNA strands together on the N-glycosidic bonds. These N-glycosilic bonds are the ones holding nitrogenous bases to the backbone, and this leads to depurination. So now we've looked at all the mutagen categories, that is chemical, biological, and physical. It's finally time for active recall. At this point, you can pause the video and get a clean sheet of paper to write down what you remember so far. The timer will alert you when it's about to switch to answers. And here we go, these are the answers to the questions. Please feel free to give feedback on both the video and the answers. These are short answers, so there's definitely a lot more content that one can write to the questions. And you'll find the articles referenced down in the description. Kindly once again feel free to comment, ask questions, or request videos. And I used Canva to edit these videos. If you'd like a tutorial on that as well, I can provide that. Feel free to also join our community on Reddit and Substack. Hope to hear from you and have a lovely time. Bye.